Our first speaker in this session is Dr. Joe Hill, who's coming forward.
All right, well, it's my pleasure to present. It's been a long 3.5 days, wonderful 3.5 days, and of course, like everybody, I'm grateful to the organizers for inviting me. I'm going to tell you a story about inflammation and several different tales about inflammation. Back in the 20th century, 1997, a very prescient individual said that inflammation can drive cardiovascular complications. That was a theory, a hypothesis, but might be a glimpse of the hidden side of the moon. And as you're all aware, inflammation-targeted clinical trials failed in numerous instances. Anti-TNF-alpha, many different trials failed miserably. In fact, inflammation was viewed as a potential contributor to a wide range of cardiovascular conditions, but all this was theory until people focused on atherogenesis, and in particular, as you're aware, atherogenesis occurs over the course of decades with the accumulation of lipid-laden plaque, but toward the end, when there is plaque rupture, plaque erosion, and acute coronary syndromes, this is where the first trial that showed for the first time ever benefit in targeting an inflammatory pathway, and that was my friend Paul Ridker, who published the CANTOS trial in 2017 with a surgical strike on a single pathway, the interleukin-1 beta pathway, and demonstrated for the first time ever mortality benefit targeting an inflammatory pathway. So then attention turned to the more decades-long process of atherogenesis. The next trial that showed benefit is called COLCOT, using colchicine targeting that very same pathway in individuals who are at high risk post-MI with elevated high-sensitivity CRP, and for the second time now, targeting that same pathway, mortality benefit was seen. Now taking it down another notch in terms of patient risk, the LODOCO trial and the LODOCO-2 trial, between them both showed, again, targeting that same pathway, mortality benefit now in patients who are somewhat lower risk. So this pathway, which initiates with the NLRP inflammasome down through IL-18, IL-1 beta, IL-6, and so forth, has emerged as a bona fide mechanism and therapeutic target in atherogenesis. Here on the right are targeting elements that have been shown to be beneficial, and here on the left are others that are being considered. In fact, there's a lot of interest nowadays on IL-6. Now, not all trials in the modern era have been successful. Methotrexate, which as you're aware is more of a sort of a global dialing back of the inflammatory response, here showed no benefit. So that's tail number one, the IL-1 beta pathway. Number two, clonal hematopoiesis of an indeterminate potential, with which you're all familiar. We haven't talked much about that in this meeting, but as we age, hematopoietic stem cells, circulating hematopoietic stem cells, they divide, of course, and every once in a while a mutation will occur that gives a selective growth advantage to one, and that clone will grow up and begin to overpopulate the immune population in the cell, in the bloodstream, and by the time we're 70, about 10% of people at the age of 70 have CHIP. They have a clone, of which there are several, culminating in this overpopulation. And what we know is that CHIP is associated with atherothrombotic risk. That's pretty well established. And now there are two mechanisms. Here is inflammatory tail number 2 and number 3, either through epigenetic mechanisms, number 1, or number 2, through neutrophil extracellular traps. These two areas are subjects of active investigation, but these are inflammatory tails number 2 and number 3 that clearly participate in cardiovascular disease and will likely emerge, I believe, as bona fide therapeutic targets. Here's tail number 4, the so-called cGAS sting pathway. You're all aware that the cytosol of a cell is not supposed to harbor double-stranded DNA. The double-stranded DNA is supposed to be packed away in the nucleus or in mitochondria. If it's in the cytosol, we all evolved to believe that that cell has been infected by a double-stranded virus, and we're going to kill it. And so there is a pathway where a molecule called cGAS recognizes cytosolic double-stranded DNA, which again shouldn't be there, and triggers a pathway that I won't go into all the details of, but culminates ultimately in a transcriptional pathway leading to interferon activation and ultimately destruction of that DNA-infected cell. In recent months, about within the last, I guess, year now, it's been shown that CHIP actually activates this CGAS-STING pathway. There's inflammatory tail number 4. That CHIP not only activates neutrophil extracellular traps and epigenetic mechanisms, but also appears to activate this exciting pathway called CGAS-STING. So there's inflammatory tail number 4. With respect to the CGAS-STING pathway, I'll digress to say that we worked on this in our lab for some time now. We posited that the cardiac myocyte probably has more DNA in it than any other cell type in the body. You're aware that 40% of the biomass of a cardiac myocyte is mitochondria, full of DNA. So the DNA, in the context of lysis, when you have an acute myocardial infarction and those cardiac myocytes are ruptured, DNA is flying all over the place. This is a hypothesis, and we hypothesized that DNA might be engulfed by mitochondria, and that might trigger this CGAS pathway in mitochondria. That was in macrophages. That was a hypothesis. In other words, the hypothesis was that DNA in the setting of an acute coronary syndrome might be an alarmin. Now, as by way of background, I'll remind you that when you have a myocardial infarction, there's a several-day period when so-called LY6C high monocytes, healing monocytes and macrophages emerge to try to well, first of all, the killing myocytes emerge first to try to clean up that mess, and then a few days later, the LYC6 low monocytes, the healing monocytes and macrophages emerge. So this first phase occurs over the course of 3 or 4 days, followed by another wave of different macrophages. So we hypothesized that cytosolic DNA sensing in macrophages might have some role in this switch between the killing macrophages and the healing macrophages. That was a hypothesis. So one of the first things we did was we asked if you have double-stranded DNA activating CGAS, this pathway that I showed you briefly ultimately culminates in these so-called interferon stimulatory genes. This is the business end that tells you that this pathway is activated. The first question we asked, is it present? And in fact, if you look at each of those 4 different molecules in the infarct zone, you can see that it's progressively increased in abundance over time and not so in the uninfarcted remote zone, suggesting that this pathway is in fact activated in that context. So then, again, as I told you, the killing macrophages come in for a few days and they're replaced by the healing macrophages. And we posited as a hypothesis that perhaps cytosolic DNA sensing through CGAS may govern this transition between these 2 phenotypes of macrophages. And in fact, if we inactivate the gene coding for CGAS and CGAS knockouts, you can see that the M2, the healing macrophages, at 3 days are more abundant. They come in earlier. You can see they're more abundant at 3 days than in the wild-type context. And at 6 days, it's even more pronounced. So it does look like we are bringing in those healing macrophages a little bit earlier. If you look at the myofibroblast abundance, it appears that the tissue is healing faster. There appears to be an angiogenic response as well. And if you look at tissue integrity using 3 different stains here blown up in a high-resolution image here, you can see that the tissue does appear to be more robust in its structural integrity when you remove the killing macrophages early and replace them with the healing macrophages. The decline in contractile performance that we're used to seeing with myocardial infarction, in fact, is attenuated when we silence CGAS. And in fact, you know that mice often suffer from ventricular rupture in the setting of MI, and in our hands we see about a 50% mortality at one week, and that is nearly completely abrogated when we silence CGAS, and again, bring in the healing macrophages earlier. Does this have any relevance to the human case? These are heart failure samples from human beings, and you can see that CGAS or STING is present, not so in normal LV, so at least it's present in the injured LV. And in fact, if those hearts are rested with mechanical circulatory support with an LVAD, you can see at least the abundance of that pathway is diminished, suggesting that there may be some role of healing here. So this is something that we now are pursuing. The postdoc who did this work is now an associate professor with tenure doing this work now for some years, a few years now. And as the prospect potentially of translating this, there are drugs that have been approved for human use that target this pathway. So we believe that it's potentially the case that this may be an inflammatory tail number 5, that this pathway is involved in pathological remodeling, at least in myocardial infarction. We're looking at other forms of disease-related stress. Now, the final inflammatory I'll tell you about, we're all familiar, we've seen these maps all over the place, that the abundance, the prevalence of obesity in our world is vastly expanding in a pandemic fashion. And it's not just in the United States, but in fact, it's around the world as projected here. And where obesity lives is where type 2 diabetes lives. We are now seeing type 2 diabetes in elementary school. It's exploding around the world. And if you take 500,000 people with type 2 diabetes, fully 27% of them have heart failure. And the type of heart failure they have is heart failure with a preserved ejection fraction, HFpEF, that we've talked about a few times during this meeting. HFpEF is sadly increasing in prevalence in our world. Fortunately, HFrEF is going down. There's now more HFpEF on this planet than there is HFrEF. And so, in work that I'm not going to discuss in great detail, we've heard about this two-hit model that we developed. We did something very simple where we decided to make a mouse that looks like a human being with HFpEF. The vast majority of people with cardiometabolic HFpEF are obese with metabolic syndrome and sometimes diabetes. The vast majority of them have hypertension. So we simply made a mouse like that that has pressure stress from a modest hypertensive intervention and metabolic stress from high-fat diet. And suffice it to say that this two-hit mechanism that we've heard about again a little bit here and there during the meeting, mechanical stress, about 40 millimeters of systolic and diastolic blood pressure, and high-fat diet, culminates in inflammatory tail number 5, so-called meta-inflammation, where upregulation of INOS is a cardinal feature of meta-inflammation that we have found leads to a superabundance of NO, nitric oxide, that covalently couples many different proteins, including the endonuclease IRU1 alpha, which is involved in splicing XBP1S, that we have found in work that we published a few years ago now, is a brake on HFpEF. This work was done in mice. We corroborated it in the ZSF1 obese rat and also in human beings from Johns Hopkins. So how is it that XBP1S is a brake on HFpEF? In work that I won't have time to show you, suffice it to say that XBP1S is a brake on this bad actor called FOXO1, which does many bad things in the heart, not least of which is a steatotic response. And you can see that we found that the way in which XBP1S is a brake on this bad actor is at the level of proteolytic degradation of FOXO1 by governing the transcriptional activation of an E3 ligase that targets FOXO1. Now, the last story I'll tell you very briefly is that you're all aware that the myocardium is an omnivore. It has to consume substrate 24/7, whether it's amino acids or lipids or glucose or ketone bodies, 24/7. It turns those substrates into ATP, and that ATP is burned for contraction. The human heart, which weighs 11 ounces, generates 6 kilograms of ATP every day, so it has to eat all the time. And in the setting of mechanical stress, hypertrophic stress, the heart will tend to use more glucose, but not if it is insulin resistant because it's been exposed to high-fat diet. So these poor hearts here are in a bind. They're being told to use this other substrate preferentially, but yet they cannot do that because of the insulin resistance that has been imposed on them. And in fact, if you look at the mitochondria in these hearts, you can see both structurally and ultrastructurally they are perturbed substantially. And if you take a dive into a mitochondrion, this is a schematic of a mitochondrion, we find that the TCA cycle is normal, as is the electron transport chain, but we've uncovered two different defects, one at the level of pyruvate dehydrogenase, which governs pyruvate intake into the TCA cycle, and number two enzymes in the beta oxidation pathway that are hyperacetylated. The deacetylase that normally cuts those acetyl groups off, SIRT3, is downregulated, as is its required cofactor NAD+. And in fact, in our model where this two-hit mechanism of pressure stress, mechanical stress, and metabolic stress, again, which I'll emphasize, hundreds of millions of people on this planet carry these two comorbidities, culminates in HFpEF, where we find, in terms of the metabolic events, we find that NAD is downregulated. There's a toxic hyperacetylation response, which diminishes mitochondrial function. If we restore NAD levels, either pharmacologically or by promoting its biosynthesis, we find that NAD levels, unsurprisingly, go back up, that hyperacetylation response is mitigated, and mitochondrial function is improved. So we've now launched our first-in-human clinical trial at UT Southwestern, targeting these patients with HFpEF and repleting their NAD levels. So stay tuned for that in the future. So with that, I will end my discussion here. Again, these types of meetings are very exciting for all of us, where the constant evolution of cardiovascular disease, just as we've seen many successes over the years, the face of cardiovascular disease today is different than it was 40 years ago. It'll be different again, and I will submit to you that the global scourge of obesity and hypertension will contribute in many ways to that. And the solution to this, even as we take care of these patients, the solution is at the level of gleaning underlying mechanism, which is the theme of this entire meeting here. So here's a picture of my lab from a little while ago. Here's Tom Gillette, with whom I worked for a long time. Here's Gabriela Schiattarella, who took on that high-risk HFpEF project, who's now been in Berlin for a year and a half running his own lab. Here's Amanda Tong, who launched her own lab. She is the MD PhD cardiologist who did the mitochondrial work and the work that I haven't shown you about sexual dimorphism, and also a program on atrial fibrillation, which I haven't had time to present either. So with that, I will stop and be happy to entertain questions if there's time.
Yes, there's time. I start with a quick question, Joe. So could you just remind us, could you just remind us in the HFpEF model where you said that PDH was changing, in what direction is it change, and what would the prediction be in terms of what would happen to pyruvate levels?
I'm sorry, what is
in the HFpEF model where you said there was a change at the level of PDH, pyruvate dehydrogenase, is it one that would predict decreased flux?
Yes, absolutely right, and I didn't make that clear, that the PDK4 is active, which suppresses PDH activity, and we're working on that actively right now. Yeah.
Go ahead.
So, Joe, I wanted to go back to that study you showed regarding fat accumulation through XBP1 and FOXO stimulation. So I just wanted, so this is a mouse model where you're giving high-fat diet to mice, right? So high-fat diet directly stimulates FOXO1. So do you think that pathway is needed when, just a high-fat diet that is given to mice can directly activate FOXO1 when that's a complicated pathway it has to go through to activate FOXO1?
Yeah, so you're right, but it turns out we find that in the context of the two hits that whole response is completely amplified.
Right.
And turns out we've studied FOXO1 in many contexts, and so it promotes a steatototic response is well established, but its activation is uniquely amplified when its degradation is suppressed by downregulated XBP1S, turns out.
Okay, so it's both through high-fat diet activation and also decreased degradation through XBP1, you would say, right?
Yes, yeah, I guess so. I don't know if the FOXO, we haven't tried to figure out if the FOXO1 is directly activated by the high-fat diet.
Yeah, that has been shown before.
Yeah, that's true, but I don't know the extent to which that might be contributing here. I don't know the answer to that.
All right, thanks.
Thanks very much, Joe, and thanks for keeping us so far in time.
All right, so far.
Thank you.
The next speaker is Lauren Field from the University of Indiana, Indiana University School of Medicine, and he'll speak about genetic variants impacting cardiomyocyte cell cycle activity. Lauren.
I would also like to start out by thanking the organizers for the chance to be here, and it's much, much, much nicer to meet old friends and make new friends in person than it is to do this stuff by Zoom. I think the only downside is that I have to keep my pants on. My lab has been interested in many years in looking at myocyte cell cycle activity, and the system we use relies on a transgenic reporter mouse that uses Jeff Robbins' alpha-myosin heavy chain promoter to drive a nuclear localized beta-galactosidase reporter. We can identify myocyte nuclei in tissue sections simply by the presence of beta-gal immune reactivity, shown here with the red secondary antibody. If we feed the mice EDU or BRDU, we can detect that cells that are in S phase by the presence of immunoreactivity, in this case to BRDU. And if we have overlay between the two, we know that this is a cardiac myocyte nucleus that's in S phase. When we do these analyses, we'll do whatever intervention we're doing to the animal, harvest the hearts, section them, and then process them for the beta-gal immune reactivity, which gives us all myocyte nuclei, stain with Herx, which will identify all nuclei, both myocytes and non-myocytes, and BRDU immune reactivity that will give us the S-phase cells. We do this with a software called Surveyor that will take nice high-power images and then stitch those fields together into mosaics of the heart. Those files are then fed into Metamorph software, and we've modified it so that it will identify potential S-phase cardiac myocyte nuclei simply by looking for the overlay of the red signal for the beta-gal and the green signal for the BRDU. We don't let the machine make the call for us. Rather, we have the machine tell us what nuclei are potentially S-phase positive, and then we manually will validate each of them. The validation screen is shown here. It has 8 panels on it. The first panel shows the mask for the beta-gal immune reactivity. In each case, the nucleus that's being interrogated is in the center of the field, and the outline of that nucleus is projected in all the subsequent panels. The second, third, and fourth panel shows the beta-gal, BRDU, and Herx signal as it would appear through the microscope. Fifth panel over here is a color combined image, and 6, 7, and 8 are simply the same images as 2, 3, and 4, but with the gain turned up. And the idea is that the computer prompts the user to either validate that nucleus as truly being an S phase or to reject it. And once the user does that, it moves on to the next nucleus. This rapidly accelerates our ability to do these types of analysis because we're looking at low frequency events. And in this particular case, you can see there's beautiful overlay between the red signal, green signal, and blue signal here, and the user would say that this was a positive result. At the end of the day, when that heart is done being analyzed, or that section is done being analyzed, the Metamorph will then circle all of the validated S-phase nuclei so we can see the anatomical position on low power images of the heart, and it will tally the total number of S-phase myocytes and total myocytes. This is an example of the assay going here. It's kind of washed out, but I have a higher contrast image of the same field here just so you can see the shape of the heart. The border zone of the infarct is here and here. The infarct itself is down here. And the little circles are indicating the pattern of S-phase cardiac myocyte nuclei in this heart. This particular animal had a permanent coronary artery ligation and 14 days of continuous BRDU infusion. And the bottom line is that there's not a lot of S-phase activity in the myocyte nuclei. We were doing a collaborative study with Kai Wollert in Hanover, Germany, and Kai had back-crossed our reporter mice into a C57 black 6 background, and we observed with Kai that the level of S-phase activity was much higher. This is an example of a heart that was 3 generations back-crossed into the B6N background, and we can see now that there's many more cardiac myocyte S-phase nuclei post-injury than we had in the D2J mice. We did some fairly simple genetic experiments with the idea of trying to map the gene or genes that are responsible for this. The data is shown on the next couple of slides. This is the values that we obtained for D2J mice that were infarcted, and all the assays are the same. It's a permanent coronary artery ligation, 14 days BRDU. The average value in these animals was, was 0.056% of the myocytes being labeled. We then took the DBA mice that had the NLAC reporter on it, crossed them into the black 6N background, and we looked and observed an average value of almost 0.8%. There's quite a range here for the values in these infarcted hearts. You have a similar range in the DBA mice. It's about 5-fold for each case. You just can't see that range because the scale is so compressed. This experiment told us that whatever gene or genes are responsible for this phenotype are acting in a dominant manner, and it also suggested that if we were to simply back-cross these F1 mice into DBA again, we should get an estimate of how many genes were involved. When we did that experiment, we saw that roughly half of the mice had S-phase levels similar to the F1 animals, and half had S1 levels similar to the D2J animals. This suggested that one or perhaps two genes were responsible for the phenotype. So we went ahead, prepared DNA from these animals, performed SNP analyses to try to look for retention of heterozygosity of a B6N allele in the segregating animals, and ask whether or not that would segregate with S-phase activity. That data was plugged into a QTL analysis, which is shown here, and we can see that there was a single highly significant peak for correlation of the retention of the black 6J chunk of chromosome and the high DNA S-phase, or the high cardiocyte S-phase activity post-injury. This region is on the distal arm of chromosome 3 in the mouse, and it was about a 15 or so megabase fragment based on these initial analyses. We performed additional backcross experiments with the idea of trying to further refine this region. The region is shown down here, the distal arm, and our original region of interest is shown in the large red fragment here. We selected 3 SNPs that were at the proximal, middle, and distal part of the region of interest, and we simply generated about 500 more backcross animals and screened them to see if we could detect crossover events. The crossover events were simply identified by the loss of heterozygosity of either one or two of the SNPs in the analyses that we did. Of the 463 mice that we generated, 53 had crossover events. We infarcted them all. 43 survived the infarct, and 17 exhibited elevated S-phase levels. SNP analysis on those guys reduced our region of interest down to 4.5 megabase pairs. And we took the animal that marked the 5' and proximal and the 3' distal border of the region of interest and performed whole exome sequencing on that, and we were able to reduce that down to a manageable 2.5 megabase fragment. This fragment harbors 13 protein coding genes, 21 non-coding genes, and 6 unclassified genes. Next, we did RNA-seq on infarcted black 6N versus D2J animals, and within this region of interest, only one gene was differentially expressed, is a gene called TNNI3K. This was identified as a negative modulator of cardiomyopathy by Doug Marchuk's group at Duke. And we also looked at black 6 DBA mice within this region by whole exome sequencing to try to identify any variants that might have predictable significance. One of those variants that we identified, there were only 3 that we could identify that had it. One was in the TNNI3K gene. This is a snippet of splice junction that leads to a frameshift and loss of function in DBA mice. And it also, the frameshift causes nonsense-mediated decay, hence the decreased expression of the gene. 2 other genes, FPTGT and ERIC3 were identified, and we ranked these genes based on the thought of what may or may not be likely to be contributing to the phenotype. Fortunately for us, Doug Marchuk, when he was doing his initial experiments, had generated a transgenic mouse where he used the human TNNI3K cDNA under the regulation of the alpha-myosin promoter again, and generated these animals in a D2J genetic backgrounds. He still had frozen sperm from these animals, so he shipped the sperm up to us. We recovered the lines and crossed them with our NLAC mice. The mice that did not inherit the TNNI3K transgene, this is in a DBA background. It looks just like the hearts that I showed you before, very few S-phase myocytes post-infarction. The transgenic mice, when we infarcted them, had now very high levels of S-phase activity. Quantitation is shown over here. So these data indicate that TNNI3K is probably driving the phenotype that we observed in the F1 animals. The assay that I showed you thus far does not distinguish the fate of these cells. It only tells us that they're S-phase positive. So the next step was to figure out whether or not the nuclei divide or if they persist as polyploid nuclei. To do that, we used inbred DBA mice or black 6 mice, gave them infarcts and fed them EDU rather than BRDU. The hearts were harvested, the cells dispersed with retrograde collagenase, and we fixed the cells to slides. Fields containing EDU positive cardiac myocytes were imaged, and we went to the Herx channel to look at DNA content. We took 15 planes of section at 2-micron steps along the Z-axis, traversing the entire nucleus, and we used that data to quantitate the nuclear content within each S-phase positive nucleus. Oh, sorry. We also looked at the EDU negative cardiac myocyte nuclei and the non-myocyte nuclei present on the same slide. After that, the cover slips were removed and we stained the slides for beta-gal immune reactivity just to confirm that the nuclei that we thought were present in cardiac myocytes were in fact in cardiac myocytes via the beta-gal immune reactivity. So when we do these types of experiments, what we observed is shown here. This is the data from the D2J heart. This is from a B6N heart. In the first column, we're looking at the EDU negative non-cardiac myocyte population. The DNA content distribution is very tight in these nuclei, and we figured that these cells are probably, or these nuclei are probably 2N, they're non-myocyte nuclei, they're not known to exhibit ploidy, and we set this as our normalization point. When we looked at the EDU positive mononuclear cardiac myocytes, we saw that on average there was a 2-fold increase in the DNA content as compared to the non-myocyte content. This indicates that the vast majority of the cells that we're analyzing, or the S-phase cardiac myocytes, mononuclear cardiac myocytes, were going on to polyploidy, and the nuclei were not dividing. Same story in the binucleated cardiac myocytes that were EDU positive. The DNA content was 2N. As we would expect, the EDU negative mononuclear and binuclear cardiac myocyte content had a 2N content as well. So what we're seeing here is that most of the S-phase events are terminating in the formation of a polyploid nucleus. If we look in a black 6 heart, you pretty much see the same story. Again, the non-cardiac myocyte EDU negative population to mark the 2N nuclei. The S-phase positive mononuclear cells, cardiomyocytes, and the S-phase positive binucleated cardiac myocytes, again forming polyploid nuclei. We can't rule out that some of these cells are not, or nuclei are actually 2N. It's just by the nature of the assay that we're doing, and it's kind of unsavory just to draw an arbitrary line in these data to say that above this threshold you're 2N and below it you're 2N, above it you're 4N. So we asked the statisticians at our institute if they could come up with a logistic model which would simply predict the probability of being 2N in these experiments, and that data is shown here. As we'd expect, the probability is high that all of the non-myocyte EDU and negative nuclei are in fact 2N, and it's very low in the S-phase positive mononuclear and binucleated cardiac myocytes for most populations. So this just gives us a somewhat less subjective way of trying to quantitate things. So the summary of this experiment was that in a F1 animal, we have about a 15-fold increase in S-phase activity post-infarction as compared to a inbred D2J mouse. Genetic and transgenic analyses indicated that TNNI3K mediates the increase in S-phase activity post-infarction, and the vast majority of these S-phase events are resulting in the formation of polyploid nuclei. We can actually quantitate those numbers here using that predicted probability threshold of 20% with the logistic estimate for the probability of being a binucleated cell that I showed on the previous slide. So this takes us off the hook for a little bit for drawing an arbitrary line, and it gives us a way to look at the data. The myocyte aficionados in the room will know that this result is at odds with the study that came out of Henry Sukov's lab and was published in Nature Genetics about 5 years ago. The salient differences between the two studies are shown in these two panels. When Henry looked at TNNI3K negative mice, this would be sort of equivalent to our DBA mice, he saw higher levels of S-phase activity post-infarction as compared to TNI3K expressing animals. He also saw that the bulk of the S-phase positive nuclei in the EDU positive population from his study in a TNA3K minus animal were diploid rather than polyploid. Henry and I are old friends. We exchanged data. We went back and forth on this. He's in the process of submitting another study that will follow up on his, and once that comes out, we'll be able to see if we resolved our differences yet or not. Finally, I just want to make 2 points from our study, which were somewhat surprising to me when we got the results. The first is that the regional analyses of these hearts suggest that the intrinsic potential for myocyte reentry is not that different between the D2J and the F1 mice. To accomplish that, we parsed the heart out into different sections. This was accomplished by drawing a line manually between the healthy myocardium and the infarct, here and here. We then asked the computer to expand this line 500 microns above and below, and to parse out that area as being the border zone. That's shown over here. Once the border zone is parsed out, it's easy to parse out the infarct zone and the surviving ventricle zone. When we look at the S-phase activities in these different zones, we see that in the remote myocardium, this is the 15-fold difference I was telling you about before between the F1 mice versus the DBA mice. Surprising to us, the level of S-phase activity within the infarct in D2 animals versus the black 6 animals was not statistically different. Moreover, the level of S-phase activity in the remote myocardium in the F1 mice was not statistically different than here. So this says in the most hostile environment that you have in the infarcted heart, the ability of these cells to reinitiate S-phase activity is actually equivalent and probably TNNI3K independent. The observation that we had elevated levels of S-phase activity in the remote myocardium was a little bit surprising as well, but TNNI3K is known to be a negative modulator of cardiac stress, and it could simply be that mice that are expressing TNNI3K have an expanded zone of stress post-MI, and as a consequence of that, we're seeing an expanded zone of injury-induced cell cycle entry. My very last slide is meant just to be a little bit provocative, and it's based on the notion of trying to predict what this level of S-phase activity might translate to. And I'm running a little bit late on time, so I just want to run through what we try to do here. We use the compound interest law, which is what your bank uses to calculate your interest, to try to predict the increase in myocardial mass over time. I thought this was an original idea until I went to the literature and I saw that this was used 125 years ago to model growth in plants. We measured what the survival at the border zone was in the border zone of our mouse models post-MI, and it was about 48%. This is what you'd expect based on the way we're defining the border zone. And we're assuming that the cell cycle rates are constant over the temporal window that I'm going to show in a prediction, and that the cytokinesis occurs within 24 hours of S-phase. And this is probably a liberal interpretation. But using this, you can kind of estimate what would happen if you only had the myocyte nuclei that were 2N were able to undergo cytokinesis, or what would happen if all of the S-phase myocytes were undergoing cytokinesis. So now on the last slide here, we see that if you have 0, 1, or 2 copies of TNNI3K, if you're only looking at the cells that are 2N and BRDU positive, there's absolutely no impact on infarct size over time. We start at 48% survivorship. You might get up to 51%, something like that, after 1,200 days of projection. If we look at all S-phase positive nuclei being diploid or polyploid in a TNNI3K null animal, we see that the story doesn't change that much. And this doesn't really bode well for the notion that there is cardiac regeneration in mice that are TNNI3K negative. However, if you look at animals that have a single copy or 2 copies of the TNNI3K gene, you actually could see complete replacement of the infarct border zone by 1,200 days in a single copy animal, after about 600 days in a diploid or a diploid mouse. This suggests, if you buy this type of analysis, that the interventions that are trying to target further cell cycle progression only, not really trying to introduce cell cycle induction, might be a viable target to promote regeneration. In other words, there seems to be an interesting reserve of cell cycle entry in these hearts post-injury, but they just can't progress through the cell cycle. And this is clearly the same in larger animals, including human hearts. If you go back and look at old histologic studies, the level of S-phase induction of peri-infarct is actually quite high. So with there, I'll stop. Sorry, I went a minute or so over. I already called out Kai and Doug's help on it. In my lab, this work was pioneered by Sean Reuter, who's a technician. Mark Sompa developed the imaging system, and I spent way too much time at the bench doing the genetic analyses. So thanks for your time.
Sorry, due to time, we're going to move to the next speaker. Can you leave your questions for during the lunch? We're over time. Thank you. Our next speaker is Dr. Ma, Professor of Emergency Medicine at Thomas Jefferson University, Philadelphia, and he's going to be talking about N-cadherin, a novel target enhancing cell therapy efficacy against ischemic heart failure.
All right, good morning. First, I also would like to start with the thanks for Jay and Ike to invite me to this wonderful meeting. And then in the past 2 days, I have definitely learned a lot. And I also would like to say that the stem cell, actually, the cell therapy is not my real field. I'm not an expert here. I'm just trying to learn. What I actually is really working on is following Abel's step and working on diabetic cardiovascular complication, specifically looking for adipocyte and cardiomyocyte communication. But when I received the invitation from Jay, I looked for the theme is cardiovascular bioengineering. I feel that probably I should, fit this better to just two small stories that we recently did, and then relate to cell therapy, and then that I'll try my best to go real quick. I don't need to convince anybody here that the cardiovascular disease remain a major problem. The acute MI, the rate is significantly reduced in past decade or two, and then but heart failure and then remain the increase. So what is the problem here that we all try as a basic scientist, and then we all try to understand what is the mechanism, how we can prevent after the MI, prevent from the heart failure. And then there are many, many things that we can all work on with these different things that in my lab also work on something. But final thing for this particular, the conference, that the cell therapy is a major focus that I just want to follow this path. For cell therapy, we all know that in the past 10 years or so that it's a roller coaster, and then that we all know that cell therapy about 15 years ago for heart failure, there was everybody feel that we can regenerate the heart, and then the heart failure is not an issue anymore. But we all also know that what happened in the past 6 or 7 years that sent us down to the ground, and then nobody really believes cell therapy can do anything, that probably make the field very, very difficult for us to publish or get a grant, because a lot of reviewers just don't bother to read detail. They just simply say, "Done work." And then, but luckily that in that very difficult time, that Circulation had the statement publication that talk about, although that is true, that the stem cells, adult stem cell, not iPS cell, okay, mesenchymal stem cells, does not develop into cardiomyocyte, but they do protect heart through different mechanisms that the independent, it's not produce new cells, but they do have secretion, every other thing. We heard a lot about this. And then luckily, a very recent study, the STREAM heart failure study, that although the major endpoint was still negative, but if you look for the last, the corner, those two figures, that you can clearly see that this single injection stem cell, and then that the mesenchymal stem cell, it does have very significant the benefit. So that promote the field, I really hope that we can come back to see this field can regenerate, the energized, and then work on this. So, but we still need to know what is the limitation here, that why after the cell therapy doesn't work. There are many, many possibilities, many issues that stem cells, they don't become cardiomyocytes, they don't stay there, and then they don't stay there long enough. So many, many things that limited these stem cell, even they don't become cardiomyocytes, even for secretion, if they don't stay there long enough, their secretion paracrine is not going to stay there too long. So that really limited the application. And also, another question, we're all in basic, in translational field, and then that we all know that basic science, we have a lot of publications find this stem cell, and then the cell therapy really works and protect heart very significantly. But clinical trials, many clinical trials don't work, and then that very minor protection. We all need to ask why that is the case. And then we all know that when in basic science, we use young animals, and then we use non-diabetic animals. We have no complications. There are many, many issues. But in clinical trials, you mixture everything together. You have age the patient, you have young patient, you have diabetic patient, you have non-diabetic patient. When you mixture them together, probably you're not going to see P less than 0.05. And then so that is one issue. And how can we improve this problem? And then from the clinical enhance or translation. From my point of view, that I feel this cell therapy is really like we need to improve three things. That we either improve our soil and then local microenvironment, or improve the seed and then cells and then or germination, that we can increase that the exosome production. So how can we do that? Our lab actually, as I said, we're focused on adipocyte cardiomyocyte communication. Many years ago, almost 15 years ago, there was a new molecule called CTRP9. That was a paralog identified in adipocyte as a paralog of adiponectin, and then that was originally discovered in adipocyte. But later on, about 10 years ago, our group was the first one, followed by other groups, identified that actually the CTRP9 expressed in cardiomyocyte even higher than adipocyte. So we start to work on this CTRP9 as a cardiokine because they produced more in the cardiomyocyte than adipocyte. Interestingly enough, and then after in diabetic patient, this cardiomyocyte CTRP9 dramatically reduced. In heart failure, after you ischemia, that the CTRP9 also dramatically reduced. And then many people already know, including our lab, and the CTRP9 is a very, very significant cardioprotective molecule. And then we ask the question, if they're produced very highly in cardiomyocyte, is cardioprotective? Probably that's a very good molecule to maintain a very healthy soil and a healthy microenvironment. If that reduce, probably that'll be very harmful to the cells if we give. So that was one study. We asked that question, that whether if we just give this CTRP9 supplementation to improve the local microenvironment, whether that'll improve injected cell and then improve their survival, improve their cardioprotection. The story was yes, and then through multiple mechanisms, ERK1, and then that paper has been published several years ago. And we also subsequently identified, so that was one study we proved that if we improve the soil, and then with CTRP9 cardiokine, we can enhance cell therapy effection. We also subsequently identified a polypeptide and a CTRP9, we call that the 281, and then that can improve germination, that actually stimulate the stem cell, the exosome production to be protective. Next question actually we asked, because this CTRP9 and then in the stem cells, that we don't know, nobody know what is the receptor. So we next ask the question, what is the receptor? If we can identify receptor, we try to answer is that whether if we know what is the receptor, we know how to improve soil, we know how to improve germination, exosome, and then if we know what's the receptor, maybe we can improve this seed, and then so that was the purpose. What we did was we utilized a mass spectrophotometer. We identified several possibilities. One of them actually caught our eye was this N-cadherin. This uncatenated, we utilized the multiple the method and then immunofluocytometry and then the precipitation that found that it does interact with the CTRP9. And if we knock out this the uncatenated, that we can significantly block the effect in vitro. So with that, we moved into in vivo, and then again, that is after we give either wild type the ADSA, or we give uncatenated knockout ADSA. And as you can see that if we knock out ADSA, that dramatically reduced the adhesion, reduced the protection. So that was published in 2017 that we proved that uncatenated is a novel the molecular, the adhesion, the receptor expressed on ADSA. And then we later on found that ADSA actually is interesting enough. It's a very interesting marker for mesenchymal stem cells, and then that form the complex with the catenin that promote cell survival. So next, that will be very straightforward. I'll keep this very quick, and then we ask the question, if we overexpress uncatenated in isolate this ADSA, whether we can improve this seed and then improve the cardioprotection. So that's the question. First thing that we did was we utilized this adenovirus-mediated uncatenated overexpression that successfully overexpressed uncatenated in ADSA. As a control, we also overexpressed T-catenated. So because we just want to know whether that's uncatenated is specific or any catenated will work. So we overexpressed the T-catenated. Surely, and then this is a method we label them and we try to track them, and then this is the result clearly, as you can see that. If we overexpress uncatenated, that can increase the ADSA retention after you inject that either 1 day, 3 days, even after 7 days, you enhance their retention. But the T-catenated does not, overexpression does not do that. So that is uncatenated specific. Also, uncatenated, that the overexpression can, in vivo-wise, we can enhance their cardioprotection, not T-catenated. That just only simply overexpression, uncatenated in ADSA that enhance the protection, but not T-catenated as a control. And mechanism-wise, and we looked for several possibilities, and then many possibilities. Finally, we identified it's 2 pathways. One is this: if we overexpress uncatenated, that increase angiogenesis, but that was expected. But something that we found interesting is also the promote cardiomyocyte proliferation, utilize different method, either the Ki67 or EDU. We found that if we overexpress uncatenated, that we can stimulate the proliferation. And then what is the further downstream that the increase is cardiomyocyte, why that does that? And then after that, we found that is in vitro-wise, if we overexpress uncatenated, we can increase this ADSA interaction with cardiomyocyte. They become stickier, so they adhere to cardiomyocyte better. And then and also through the mechanism-wise is through paracurrent. If we isolate the conditional medium, we can simulate the similar mechanism. Finally, we tried to understand what's the mechanism, and we did the analysis. There are many, when we overexpress uncatenated, there are many molecules that increased, which did the one by one. Finally, we narrowed down 2, actually 3. One is HGF, another 2 are MMP 10, 13 in ADSA. And then after that, we asked the question whether that is important. This is in vivo. We can clearly see that also increase this 2 molecular expression. And then interestingly, we found that if that for the angiogenesis, it is and also migration and angiogenesis through 2 different mechanisms. For the migration, that is mediated by MMP 10, 13, not HGF. But for the angiogenesis, that requires both, that if you knock down the HGF or the MP10, 13, it both block. But for proliferation, it's interesting. Proliferation is not affected by MMP 10 or 13, it's affected by HGF. If we knock out HGF, it blocks the effect. So we also, last question, the signaling-wise, is why this work? And then through multiple the study, we finally find that it's overexpression is intercalarin that can increase beta-catenin expression and also promote the cellular translocation. The N-cadherin overexpression can promote beta-catenin, the migration that if we knock out beta-catenin and then we block this N-cadherin effect. And then so final summarize, and then so we found is N-cadherin is a molecule that expressed on ADSC surface, and if we overexpress that, we can promote their interaction with the beta-catenin. Beta-catenin will get into the nuclear and then promote two different pathways. One is HGF that can promote cardiomyocyte proliferation and angiogenesis. Another one is MMP-1013 that promote migration adhesion that finally they can improve the cardiac protection. That has been published 2 years ago. This is the final slide. I just want to acknowledge those people still working in my lab. This study would not be possible if without collaboration with Dr. Yajing Wang. Currently, she's in UAB in Jay Zhang's lab. Thanks a lot.
Thank you. We have time for questions.
In your department.
We did exactly the same study, overexpress N-cadherin in mice in transplant cell. It showed exactly same effect, but we didn't examine the cell cycle, or we didn't really see the cell cycle increase. The beta-cadherin affect the cell cycle mechanism. That's very interesting that you presented. I also want to clarify that Emerson's work in JCC, thank you for bringing that up. It's mainly through the modulate, MSC through the regulate immunomodulation. So the patient with higher blood CAP, I think it's an indication of the inflammation. That's the patient population that works.
Yeah, very true. I really appreciate your comments that we're going to
Thank
follow
you.
that step and then discuss with you to design additional study. Thank you.
Go ahead.
Thank you very much. Excellent talk. I have one question. Have you checked connection 43? I wonder that there are interaction between connection 43 and N-cadherin.
Actually not. And then that's a very good suggestion. We can check that in future study, but in this study we didn't check.
Thank you very much.
All right, thank you.
Okay, Jima, thank you very much.
All right, thank you.
Okay, the next speaker is Maria Contaridis, and she'll be speaking about her favorite kinase, ROA, in response to cardiac injury and cardiac fibrosis. Maria.
Thank you so much, Dale. I just want to echo my colleagues' comments and just say thank you to my dear friend, Dr. Jay Zhang, and Dr. Fukada for the kind invitation to be here. I brought my daughters, and we've really just enjoyed the time here, my first time to Japan, so thank you for your hospitality. So today I'm going to talk about a novel story, although it is one of my favorites, but not all of my favorites, on the role of ROA in response to cardiac injury and cardiac fibrosis, and just some of my disclosures that are independent of this talk today. So we've heard over the last couple of days a little bit more about the mechanisms that we're starting to really understand that drive the pathological response that leads to hypertrophic cardiomyopathy, and then the chronic activation that eventually leads to a dilated cardiomyopathy and heart failure. And while we are really understanding these physiological parameters, we're still actually struggling to understand some of the complex signaling molecules that are involved in some of these processes. And just as an example here, I just want to highlight this induction that you see in PI3 kinase AKT at these early stages of cardiac stress that lead to pathological hypertrophy, and knowing that this same signaling pathway is also significantly increased in a physiological hypertrophy. So it's still not very clear how it is that the same signaling pathways are driving pathological and physiological effects. Similarly, you can appreciate that later on, once hypertrophy happens and you're moving into a chronic dilated cardiomyopathy, the same signaling pathways are decreased. And so we really still don't understand why those signaling processes happen this way. Similarly, we know that the overt overexpression of my favorite molecule, RhoA, also has been shown to demonstrate a very pathological dilated cardiomyopathy and heart failure very early on after birth. Joan Heller-Brown's group also showed that a more subtle overexpression of RhoA can lead to a protective effect in contrast. So there they saw a decrease in scar tissue formation and enhanced cardiac function and decreased fibrosis. And so to add to the complexities of this story, we actually previously also showed that when you delete RhoA, you actually see a very similar phenotype to the overexpression of RhoA, where you saw an increase in the progression of dilated cardiomyopathy. In contrast, however, there was also a cardioprotective effect in these mice, where we saw that there was a significant reduction in the development of fibrosis. And so we're beginning to understand why some of these dichotomies exist within the heart and within tissues. And we think this has to do with the multiple different cell populations that play a role in the development of heart disease in patients. And so what is RhoA? It belongs to the Rho subfamily of GTP binding proteins, best known for their regulation of the actin cytoskeleton. And they're involved in actin stress fiber formation and adhesion complex assembly, especially in fibroblasts. And we know that its expression and activity are induced by hypertrophic stimuli, whereby activation by agonists that induce hypertrophy, including endothelin and PDGF, can activate RhoA by converting its inactive GDP isoform to an activated RhoA isoform with GTP bound. This then leads to the activation of its downstream effector, Rho kinase, which then modulates a series of downstream signaling effects that can control things like contractility, calcium regulation, proliferation, and cell death. And like we discussed a little bit earlier in the last couple of days, we and others have studied this primarily in the context of cardiomyocytes, endothelial cells, and fibroblasts, but not much has been known about the many other cell populations in the heart that primarily affect contributions to development of heart disease, and this includes the immune cell population. So we asked the question, does RhoA actually modulate cardiac injury and inflammation in the heart? And we started by looking at the effects of RhoA expression in response to myocardial infarction. So after 4 weeks of infarct, we isolated both non-infarcted area and infarcted area, and we saw that indeed both infarcted and non-infarcted areas at 3 and 7 days post-infarct were significantly increased for the expression of RhoA. And so when we looked at the single-cell RNA sequencing level, we could see that RhoA actually is expressed in multiple different cell types within the heart. And when we remove the primary signaling or the primary expression in the cardiomyocytes to look at the other populations in response to cardiac infarct, we found that indeed these were not only differentially expressed in 24 different cell types within the heart, but that they were multiple different cell types expressed at different time points within the myocardial infarct, suggesting that it's not just the cell type that's really integral to the development of heart disease, but it's also the timing of when these proteins are expressed. So we know that macrophages play an important role in both inflammation and tissue repair. And while this is a heterogeneous population within the heart, you can basically classify this as 2 different populations, the CCR2 negative macrophage and the CCR2 positive macrophage. So basically, the CCR2 negative macrophages are an embryonic developmental origin, so they're resident cells, whereas the CCR2 macrophage are primarily infiltrated in the heart through monocytes. And the important thing about these cell types, as Joe Hill just talked about a little bit earlier, is that in response to stress, these cells become activated. And whereas the CCR2 negative cells have phagocytic properties, their primary goal is to actually help regenerate the heart and create that healing property. whereas the CCR2 positive infiltrating cells are the primary inflammatory mediators that drive reactive oxygen species and production of cytotoxic cytokines within the heart. So importantly, as I mentioned earlier, timing is really important. And in response to myocardial infarct, we know that the resident macrophages actually undergo a very significant apoptotic cell cycle, which means that then the infiltrating CR2 positive cells move into the heart at this very early stage and induce this hyperinflammatory response that leads to activation of these cytotoxic cytokines. By the time this thing starts to lay off and the macrophages start to die out, we repopulate the CCR2 negative cells, the resident cells, that then take over at around day 7 after the myocardial infarct. And these lay the groundwork for activation of fibroblasts, which lay down the collagen that leads to scar tissue formation. And while that seems like a deleterious effect, that actually has healing properties within the heart in response to a myocardial infarct. So we wanted to ask, how does RhoA specifically modulate monocyte macrophage regulation during cardiac repair? And so we looked back at our distribution table, and what we found was that in addition to cardiomyocytes, endothelial cells, and fibroblasts, as we showed earlier, have significant responses in the heart, RhoA also has a very significant effect in both CCR2 positive and CCR2 negative macrophage, in that the timing of the expression of RhoA in these cell populations is actually differentially regulated. So CCR2 positive cells express RhoA very early during that massive infiltration time period at day 3, whereas CCR2 negative cells take over and hyperexpress RhoA at day 7 when the CCR2 negative cells start to take over, suggesting that RhoA can have these differential properties in different cell populations within the same macrophage. And this is actually validated translationally when we look at what happens in humans in response to dilated and hypertrophic cardiomyopathy, which is a much more progressed part of the disease, where you can appreciate, although that macrophage, the CCR2 positive cells, still express RhoA, the majority of RhoA is expressed in the CCR2 negative proliferating macrophage that are already in the resident tissue of the heart. So in order to try to understand more specifically how RhoA is making an effect in myeloid progenitor cells, we decided to create a myeloid-specific deletion of RhoA crossing flox RhoA mice to the lice 2 Cree. And what we found was that after 4 weeks of myocardial infarct, the hearts in the RhoA deleted myeloid cells had significantly increased infarct size. Moreover, using micro-CT scanning, we could appreciate that these hearts were actually significantly more dilated than the wild-type counterparts. And this was quantified here to the right. And what was really interesting about this study, actually, is that there was a sex difference between these animals, where we did not see any significant effects of RhoA deletion in the myeloid lineage in the female mice at this age, suggesting that these mice are probably protected hormonally from the development of cardiovascular disease as mediated through RhoA. So for the rest of my talk, I'm only going to be focusing on male mice, given that the female mice did not have any significant differences. And so, as expected with a more severe cardiac phenotype, we also saw that these mice had significantly increased heart weight to body weight ratios and heart weight to tibia length ratios, as well as decreased cardiac function, where we saw that ejection fraction, fractional shortening were both significantly decreased compared to the wild-type myocardial infarcted mice, and that the left ventricular mass and the stroke volume in these animals was significantly elevated, suggesting that the deletion of RhoA in the myeloid lineage alone was sufficient to progress cardiovascular disease in these animals. And as suggested by the increased and enhanced scar size, you can appreciate that this also led to an increased size of, or increased amount of fibrosis in these animals, which was also further quantified by looking at RT-PCR for fibrotic factors such as CTGF and collagen, which again were all significantly increased in the myeloid-specific deleted population. So we know so far that deletion of Rho A in the myeloid lineage leads to a worsened cardiac function, increased scar formation, and decreased cardiac function. So the next question we asked was, is this worsened phenotype mediated by an increase in the influx of the pro-inflammatory CR2 positive monocytes? And much to our surprise, when we actually looked at the number of monocytes in the heart tissue of these animals, we found that there was a significant decrease. And when we further fax analyzed these, looking for surface markers of either CCR2 positive versus CCR2 negative, we found that while the CCR2 negative population was largely unaffected in number, the number of CCR2 positive cells was significantly decreased. So this was kind of a surprise to us, given that these are the inflammatory cells that would lead to an enhanced myocardial dysfunction in these animals. So we did an additional experiment to validate these results using MAC3 antibody staining to actually stain the macrophage within the heart tissue. And again, you can appreciate that the deletion of Rho A in these myeloid lineage cells had significantly less expression of the MAC3, suggesting fewer macrophage in the heart than in the wild type. So what could this be a consequence of? So if you have less CCR2 cells within the heart, it means one of two things. Either you're producing less monocytes in the bone marrow and releasing them through the spleen, or there's some problem with honing in the circulation. So to address this question, we actually looked at the number of monocytes within the spleen, and as you can see from these data, we did not see any significant difference in the number of monocytes that are produced, suggesting that Rho A has no effect in mediating the production of the myeloid lineage. However, when we looked at the number of cells, of myeloid cells in circulation, we found that the Rho A deleted myeloid cells within the blood were actually significantly higher than the wild type cells in response to myocardial infarct, suggesting that the effects that we see from deletion of Rho A in this lineage have specific consequence on the homing of these cells to the area of infarct. And so because it's a homing defect, it could be one of two things. Either there's a problem with adhesion of these cells to the scar tissue area, or there's a problem with migration of these cells and infiltration into the scar tissue area. And so to be able to delineate these two things, we actually did first an adhesion assay, and what you can appreciate is that both at 30 minutes of LPS stimulation of bone marrow-derived macrophage to induce the activation of these cells, and 60 minutes, you can see that there was significant decrease in the adhesive properties of these cells compared to the wild type. Moreover, when we looked at the surface expression of adhesion molecules on the monocytes, we could see that there was a significant decrease in both VLA4 as well as PSGL1, which are both markers for adhesive properties on monocytes into the infiltration areas. They were both significantly decreased in the myeloid-specific deleted cells. Moreover, when we looked next to see if there was a significant effect on migration of these cells, we actually looked at baseline and saw no differences, but in response to CCL2 stimulation, which is the driver that leads to the infiltration of monocytes into the infarcted area of injury, you can appreciate that, again, there was this very significant decrease in the ability of these cells to infiltrate into the area of disease, suggesting that in Rho A-deleted myeloid cells, there is a significant defect in the ability of these cells to not only migrate and adhere to these cells, but also to infiltrate and to be able to function within the cardiac tissue. So the loss of myeloid cells decreases adhesion, migration, and infiltration of CCR2 positive monocytes to the site of injury. So that's all well and good, but if there are fewer infiltrating CCR2 positive cells in response to cardiac injury, how do we get a worsened cardiac phenotype? So we asked, does deletion of Rho A affect CCR2 negative resident macrophage? And to do this, we looked at two aspects of these cells, their phagocytic properties and their ability to do lysosomal degradation. And as you can appreciate from this experiment, in response to LPS stimulation of these bone marrow-derived monocytes, there was no significant difference either at 1 hour or 3 hours in the ability of these macrophages to absorb or phagocytose debris tissue. In this case, it was a stable dye. However, when we looked over time in these cells, we could appreciate that the deletion of Rho A in these myeloid cells actually delayed or prevented lysosomal degradation, suggesting that these CCR2 negative resident cells could absorb the scar tissue but couldn't clear that debris in a timely way, suggesting that that could be a potential mechanism for the development of increased fibrosis in these animals. And to further delineate what could be happening as a consequence of these aberrant CCR2 positive, CCR2 negative effects, we actually looked to see at what happens with cytokine production. And what we found was both in the heart as well as in the bone marrow-derived macrophage, there was a significant increase in both IL-1 beta and IL-12, two very hyperactive cytotoxic cytokines that lead to cardiac disruption and increased incidence of heart failure in these animals. Moreover, when we looked finally at what signaling mechanisms might be disrupted, we're coming full circle to looking at PI3 kinase, as I brought up earlier at the beginning of my talk, where you can appreciate that deletion of Rho A in these monocytes leads to an enhanced activation of PI3 kinase signaling, as well as increased STAT3 cytotoxic downstream signaling pathways. So taken together, we have a proposed mechanism for myeloid-specific regulation of Rho A in response to cardiac injury and fibrosis, where we suggest that deletion of Rho A in myeloid cells can lead to an increase in post-infarct scar tissue and a worsened cardiac function. We think that this also the deletion increases cardiac fibrosis, and also in myeloid cells decreases the infiltration of CCR2 positive cells, suggesting for the first time that this alone is not sufficient to induce an inflammatory and increased cardiac dysfunction phenotype. We also show that the deletion of Rho A in these myeloid cells can increase the expression of IL-1 beta and IL-12 B, which are both cytotoxic cytokines, and that the deletion of or increase of these cytokines leads to an activation of downstream PI3 kinase and STAT3 signaling, which drive profibrotic signaling pathways. So we have a model which shows that myeloid-specific expression of Rho A in CCR2 positive cells is important for the infiltration, migration of these cells, whereas in CCR2 negative cells, it's important in decreasing the cytotoxic cytokines and increasing the infiltration and decreasing fibrosis ultimately. So that's the yin and yang of Rho A signaling and other molecules like that, where too much and too little are both bad because they have differential functions in maybe even the same cells over a different period of time. So there's very important, I think, to really kind of delineate some of these signaling pathways and mechanisms that lead to disease by looking and parsing out at what happens exactly at different time points and in different cell populations within the heart. So with that, I want to acknowledge the group that did the work. All of this was really done by one very talented postdoctoral fellow, Bing Su. I want to thank all of my grant support, and for anyone interested in both faculty and postdoc positions, please send me an email. We're very interested. Thank you, and I'm happy to take any questions.
Thanks, Maria. We are open for questions. I guess that was super clear.
Okay,
Thank you.
Great, thank you so much.
Okay, the final speaker in this session is Nicole Purcell, and she will be speaking about systemic nicotine on inflammation, cognition, and aging in adolescent mice. Take it away, Nicole.
Thank you. I'd like to thank the organizers too. This is going to be a little bit of different talk. It is a very new area in my laboratory, and it's looking at both the heart and the brain and the effect of nicotine on adolescent mice. And it's been great to be here and hear all the science. So again, thank you to the organizers, and I'm having a wonderful time. And so why nicotine? And so vaping has become an epidemic, and especially in the United States, more and more people are turning to vaping as an alternative to normal cigarette smoking, thinking that it's a healthy alternative. But it's not, and we know this over the last couple of years, that not only the vaping constituents, but nicotine has always been a part of that. And for the longest time, when I used to say that nicotine is bad, they would say, well, it's the addictive part of it, but it doesn't really have that many detrimental effects, and that's not true. And over the years now, nicotine is becoming more and more known with having effects not only on your heart rate, blood pressure, but it's also having these effects that we've shown on oxidative stress and inflammation. And so my lab became interested in looking at nicotine because we work on a serine-threonine phosphatase known as FLIP. And FLIP is a family of phosphatases that have two isoforms. FLIP1 alpha and beta are splice variants of FLIP1, and FLIP2 is the other isoform. They are ubiquitously expressed, so they're found throughout the cell. They are regulated through degradation and translation. At least that's what's known in cancer. When it comes to the cardiovascular or the cerebral vascular system, not much is known about the regulation of the phosphatases. We do know, however, that if you look at their structure, FLIP1 beta and FLIP2 differ by this RAS association domain. And that'll become important later when I talk in the brain, because through that RAS association domain, FLIP is not only able to interact with AGC kinases, which are important for a lot of homeostatic regulation like cell survival, metabolism, proliferation, but it's also able to work on the ERK signaling pathway, which is also involved in survival, but is very important in the brain for memory formation. So we have shown that unlike in cancer, where FLIP1 and FLIP2 are tightly regulated, FLIP1 and FLIP2 are needed for the homeostatic control of cell survival. So in cancer, these two isoforms become dysregulated, and you find that as a cancer is a higher grade, so like 4th grade glioblastoma or prostate cancer, you find that both of these isoforms are gone that lead to aberrant growth and metastatic tumors. However, what we found to our surprise is that FLIP1 and FLIP2 do not have the same targets under a more physiologic state. So we showed that when we knocked down FLIP1 in a cardiac myocyte, we saw that we changed the 473 phosphorylation, which is the hydrophobic motif, and that is where the FLIP phosphatase works. However, when we knocked down FLIP2, we saw no regulation of AKT, and that was very surprising because up until then, FLIP1 and FLIP2 were thought to both regulate AKT. And so we were able to show that there was not only a tissue specificity, but also that we were having a cell specificity. When we went to make the global knockout for FLIP1, we saw that the FLIP1 did remove all of FLIP1 throughout the mouse, and we altered AKT at the 473 site, which gave it a basal increase in AKT activity. However, this led to no cardiac dysfunction, no hypertrophy. And we saw that it was activated in the whole heart as well as the cardiomyocyte. So when we went to stress these animals, because they had this increased AKT basally, what we found was that the FLIP1 knockout had an increase in exercise-induced physiologic hypertrophy, and that if we tacked these animals to put them under pressure overload, we saw that we actually attenuated the pathologic response. They had a decreased hypertrophy, they had no fibrosis, they didn't have activation of the fetal gene, which really surprised us. What we saw was that, unlike the wild-type heart, that after 2 weeks hypertrophy, there was a decrease in the angiogenic response, and so you had a decrease in the myocyte to capillary ratio, which would most likely lead to a more ischemic damage in the heart. The FLIP1 knockouts maintained an angiogenic response and had increased angiopoietin, VEGF, and we were able to show that we have much more capillary density baseline in the FLIP1 knockout that actually led to this protection after TAC. When we went and looked at the brain, because as I said, it's a global knockout, FLIP1 is increased in the brain, and when we used a stroke model of ischemia reperfusion, middle cerebral artery occlusion, you could see the FLIP1 knockouts were extremely protected. However, if we block the AKT signaling pathway prior to injury, we could reverse that protection and see the same amount of injury in the brain as the wild-type mouse, suggesting that the protection that we see in the FLIP1 knockout partially is due to the AKT activity that we see. So why am I interested in nicotine? And so we know recently that exposure to nicotine actually allows the heart to be predisposed to injury. So recently there was a study in the rat where they just nicotine infused prior to IR injury and showed an increase in infarct. Our lab has used osmotic mini pumps to infuse nicotine for 7 days. We use 5 mg per kg per day, which is equal to someone who's smoking a high level or vaping a high level of nicotine. So the circulating systemic nicotine that we have, that when we do 1 hour of ischemia, which followed by 24 hours of reperfusion, we can see that the heart has much more damage following nicotine exposure. And so we ask the question, why? What is going on with nicotine that's allowing for predisposition to injury? We know from our studies that removal of FLIP1 is protective, and it's needed for increased survival in the heart. So we asked the question, were there any changes in the FLIP isoform due to nicotine? So we repeated the experiment where we took both our FLIP1 knockouts as well as the wild-type hearts, infused them using the osmotic mini pump with nicotine, and asked what happens to the FLIP isoforms. What we saw is that in the wild-type heart, we had an increase in FLIP1 protein expression, and we had a concomitant decrease in the FLIP2 protein. As I just showed you, removal of FLIP1 is protective. Upregulation of FLIP1 is actually found in an aged heart, and it's found to decrease AKT activity and be detrimental in an aged animal. And so we were surprised by this. In our FLIP1 knockouts, where we have no FLIP1, we actually saw an increase of FLIP2 that we think is maybe a protective mechanism, that you're getting double protection, because if we overexpress FLIP2 in a cardiac myocyte, we can actually protect from oxidative stress. And so we have both the FLIP1 and the FLIP2 knockout mice in the lab, and so we subjected them to IR to see if changing the isoforms, how that affected injury. And as you can see here, we do get some protection when we remove FLIP1 after 1 hour of ischemia and 24 hours of reperfusion. However, if you remove FLIP2 in the animal, you have much more injury, suggesting that what we saw with the nicotine, possibly, this could be due to the dichotomy of our FLIP1 and FLIP2 isoforms. And so we wanted to delve a little further to look at, before going back to the IR injury model, is to understand what's going on in the heart. And what we found was that we know that nicotine has been shown to cause ROS in cardiac myocytes, but in the whole heart, we found that there was an increase in NOx4 that I'm not showing you here by protein that was totally blocked by the removal of FLIP1. And we also saw that the removal of FLIP1 increased antioxidants such as catalase, SOD2, and NRF2 in the presence of nicotine that were all downregulated in a wild-type heart that was exposed to nicotine. We saw this both at the protein and the mRNA level, suggesting that you're having this imbalance. As we increase ROS, we need the antioxidants to decrease that. And in a wild-type heart exposed to nicotine, there's this imbalance. And so we believe that you're getting much more ROS. And so we decided to look at some of the changes in the OXPHOS proteins in the whole heart. And when we blotted for the mitochondrial complex proteins, we found that they were all downregulated in a wild-type heart following just 7 days of nicotine exposure. However, the FLIP1 knockouts either kept their OXPHOS proteins or significantly increased them following nicotine exposure, suggesting that there might be damage to the mitochondria going on following nicotine exposure, whereas the removal of FLIP1 is protecting that. So we were thinking, with the damage to mitochondria, were there any changes in autophagy? So we looked at the mTOR pathway, because AKT plays an important regulator of AKT. AKT plays a regulator of mTOR. And what we found is that when you treat wild-type mice with nicotine, you had an increase of phosphorylation of mTOR and a blocking of autophagy. However, when you remove FLIP1, we actually have an increase in autophagy, and we can see that by the activation of ULK downstream of mTOR with FLIP1 removal. So we believe that there is a difference in the ability of the cells to remove the damaged mitochondria in the wild type that removal of FLIP1 is protecting. And we have a lot more work to go into this further, but we took a step back to go into the cardiomyocyte to actually look at mitochondrial respiration and what was going on. So first, we treated the myocytes with nicotine for 48 hours to show that we also get a concomitant increase in the FLIP1 protein, as we saw in the whole heart. I'm not showing you here, but we do see a change in the FLIP2 protein. It does decrease in the cells. We also get an increase in ROS formation, so we got an increase in NOX4. We also see changes in the antioxidants, which I'm not showing for time, but we see everything that we saw in the heart with these changes. We also saw changes in the mTOR activity. So we asked the question, what does nicotine do to mitochondrial respiration? And nicotine alone decreases the maximal respiration. However, if we remove FLIP1 in these cells, we rescue that mitochondrial dysfunction and we rescue respiration. And so nicotine alone is causing changes. And so we asked, is this change from nicotine from the upregulation of FLIP1? So we took the cardiac myocytes and overexpressed FLIP1, and we looked and we saw that just overexpressing FLIP1 alone decreases basal respiration, also maximal respiration, and alters proton leak. So we think that removal of FLIP1 is preserving mitochondrial function. Also, it is decreasing the amount of ROS because of the antioxidants and protecting the heart this way. But what we really don't know, and what my postdoc is really looking into, is how is nicotine regulating the FLIP isoforms? As I told you, most of the work is done in cancer, and we don't know how FLIP is changing in the heart. We have preliminary data that we just got last week that suggests that it's probably translationally regulated in the heart under nicotine. Whether this is different for under ischemia reperfusion or pressure overload, we're investigating that now. So it's really exciting for us to understand how these isoforms are regulated and also their effect, because if we're going to target them as a protective mechanism, we really need to understand what FLIP1 is doing versus FLIP2. So right now, we believe that the nicotine is altering our FLIP expressions to predispose the heart to injury by decreasing antioxidants, increasing ROS formation, and damaging mitochondria, and also altering autophagy. That's in the end leading to increased oxidative stress and most likely cardiac injury. And when we remove FLIP1, we see a concomitant upregulation of FLIP2 that's actually, overall, we're seeing an increase in oxidation, antioxidants, and a decrease in oxidative stress, and a preservation of the mitochondria. So we think, and we're hopefully can show, because it has been shown in C. elegans, that FLIP can alter autophagy through pink parkin. And so we know that FLIP1 removal under that situation has been able to show that it leads to decreased fragmentation of mitochondria and increased lifespan in a C. elegans. So it'll be interesting in the heart to know if with nicotine exposure, if this increase in FLIP1 is altering the ability of damaged mitochondria to be removed, and you're leading to more cell death, which we show in a cell culture system, which I haven't shown here today. But what I wanted to show also was we're also looking at the heart-brain. Since we have the global knockout, and we know with aging and injury, the brain sees increased ROS that leads to impaired cognition, you have your increased brain disease, we have neurodegeneration, and you see changes in inflammation. So we know as the brain ages, there's this term called inflammaging, you get an increase of inflammation. And so we can look at what happens to the brain in our knockout animals versus the wild types after that 7-day exposure to nicotine. And we wanted to know what effect does aging have on FLIP levels in the brain. So we're looking at that over an aging time. And what is the effect of nicotine on the adolescent brain? As I said, we're talking about young people who are vaping more. 40% of high school students have vaped. And so you want to know what's happening to the developing brain. And so, as I said, in the heart as well as the brain, when we age mice, we're able to show that the FLIP1 levels are increased. And that is a corresponding decrease in AKT. As Maria presented, changes in the signaling over time and leading to heart failure. We know as an animal ages, you also see changes in cardiac function. And so we were interested in understanding, well, if aging has this increase in FLIP1 and blocking of AKT, how is nicotine that's increasing FLIP1? What is that doing in the setting of an adolescent brain? Is it looking like an aged brain? What would we see in more of like a 24-month where you see this decrease in AKT? And so we looked at the expression levels, and what we found was in the brain, just like the heart, the levels of FLIP1 proteins were significantly increased after nicotine exposure. However, different from the heart, we did not see any changes in the FLIP2 isoform. And that's really exciting for us because that shows a different regulation in the heart and the brain that we need to tease out how are these isoforms being affected by nicotine. We did see, and I'm not showing because I'm running out of time and I want you guys to get to lunch, is that we see the same changes in the brain that we see in the heart. We see increased oxidative stress from wild type brains. We see increase in the FLIP1 knockout brains, we see an increase in antioxidants. We see a decrease in reoxidative stress. And so we thought, let's go longer. 7 days is not really going to have an effect on memory or anxiety, since people who are vaping now have changes in anxiety, mood swings. So we put them on a 28-day regimen with the same amount of nicotine, 5 mg per kg for 28 days, and we did behavioral studies. And today I'm just going to show you one of those behavioral studies, the Y maze, but we put them all through Y maze, novel object recognition to look at memory. We also put them through marble bearing and dark light box to look at anxiety. And so, as I told you in the beginning, there's the RAS association domain that FLIP can interact with. And it's been shown in the brain that overexpression of FLIP blocks ERK and can cause problems with memory consolidation. So my question was, what happens since nicotine's increasing FLIP1 levels into the heart, into the brains of mice? And it's sad, but with just 28 days exposure, that under the Y maze test, and so what you do is you put a mouse in a Y maze and block off the novel arm. Allow the mouse to explore for 15 minutes. You let the mouse rest again in the cage for 1 hour. You bring them back to the cage, and they should be able to see that once you open that novel arm, that they haven't explored there. They're going to want to go there more because they should remember that they've been in the other arm. If they have no memory, they'll explore both the novel as well as the open arm in the same amount. You can see here that the amount of time spent in the novel arm was over 50% for a wild-type mouse that was only subjected to saline for 28 days. However, just giving them nicotine for 28 days, the time spent in the novel arm reduced to less than 10%. So we could see that they're already having memory problems. I'm not showing you, but they have anxiety. They're not burying the marbles as much as they should. And so we're seeing these behavioral changes in the brain. And so I know a lot of people say, well, it's systemic nicotine. How does this react to vaping? And so a colleague, Dr. Lauren Wold at OSU, is doing a lot of studies on the heart and vaping. So he sent me the brains since he doesn't look at them. And we can see a significant upregulation of FLIP1 in these brains that have been exposed to vaping. And we do see inflammation as shown by TNF-alpha and IL-18. And so we believe that nicotine exposure is taking a healthy adolescent brain and making it look like a 24-month-old mouse. We're seeing increased inflammation, FLIP1, and mitochondrial dysfunction, which I haven't shown you today, but that in a FLIP1 knockout, we reduce all of this. And so my lab's working on studies both in the cardiomyocyte-specific knockouts as well as astrocyte-specific knockouts, but because we know as we age, astrocytes become reactive and they lead to CNS dysfunction through neurons, microglia, and oligodendrocytes. So I want to thank you all for listening to me before lunch. I want to thank Kaja, who's my postdoc, who's done most of this work, and Alyssa, who's working on all the behavioral studies. And I'd like to thank my funding agencies and just throw a plug in for AHA. I wouldn't do my role as communications chair to say, I hope that you all would become members of AHA, especially the BCVS Council, and come see us all at either BCVS this summer in Boston or at Sessions in November. Thank you.
Thanks so much, Nicole. Go ahead.
concentrations of the nicotine is various.
Right.
So what's you suggest about the
So for this, we've kept it regulated at 5 mg per kg, so it's equal to like 100 nanograms of conitine in the blood circulation. We wanted it to be a high dose of like someone who, because we know in the vaping of the adolescents, one cartridge could be equal to a carton of cigarettes, and because it's unregulated. So we wanted to keep it to that, a heavy smoker, just for a short period of time. But we want to move into the vaping model next. But we wanted to try to have a clean model to tease apart, especially since we're trying to figure out mechanism of action. Thank you.
Okay, thank you very much, Nicole. And I want to thank all the speakers for an outstanding session. Also, thank you all very much for keeping us on time. So I guess there's lunch back there, and there'll be a transition to the lunchtime symposium.